an alkali. An acid is neutralized when an alkali is added to it. Here is an equal amount of acid and alkali. Let's neutralize the acid by adding a whole beaker of alkali to it. Add 3 drops of universal indicator. It looks like too much alkali is added. Let's do it again. This time, the amount of alkali solution added is cut into half. Add 3 drops of universal indicator. Now, it has become more acidic. So, what is the best way to determine the exact point where neutralization is achieved? At the end of this lesson, students should be able to describe acid-base titration, determine the endpoint of titration during neutralization, solve numerical problems involving neutralization reactions to calculate either concentration or volume of solutions.
When an acid is added to an alkali, salt and water are produced. This reaction is called the neutralization reaction. The acid is said to be neutralized when all hydrogen ions from the acid combine with the hydroxide ions from the alkali to form water molecules. But how much alkali is needed to ensure all hydrogen ions present are neutralized? This can be done through titration. Titration is a quantitative analysis method to determine the volume or the concentration of an acid needed to be neutralized by a fixed volume of an alkali. In this method, all materials must be accurately measured. Thus, a burette is used to add acid into an alkali in a measured way. The alkali then is accurately measured and transferred into a conical flask using a pipette. How can we tell the exact point when all hydrogen ions present are neutralized? The point when all hydrogen ions from an alkali combine with hydrogen ions from an acid to form water molecules is called endpoint. Since both the reactants and products formed are colorless, we can only determine the endpoint with the help of an acid-base indicator like phenolphthalein. Let's carry out an activity to determine the endpoint in titration of 1.0 mole per cubic decimeter hydrochloric acid and 25.0 cubic centimeter of 1.0 mole per cubic decimeter sodium hydroxide. Click on the button. Clamp a 50 ml burette to a retort stand. Place a filter funnel and open the stopcock. Rinse the burette twice with 5 cubic centimeter of hydrochloric solution. Close the stopcock and fill the burette with 50 cubic centimeters of hydrochloric acid. Open the stopcock to fill the tip of the burette with hydrochloric solution and to remove bubbles. Place an empty beaker under the burette. Now, the burette is ready to be used. Rinse the pipette. Withdraw 2 to 3 cubic centimeters sodium hydroxide solution with a pipette bulb. Invert the pipette to a horizontal position. Turn and tip the pipette so all inside surfaces have come into contact with the solution. Then, drain out and discard the solution. Squeeze the pipette bulb to empty out the air. Then, slip the bulb on the flat end of the pipette. Place the pipette into the solution, slowly releasing the pressure on the bulb, but maintaining a seal between the bulb and the pipette. Watch the solution rise in the pipette. Do not let any solution get into the bulb. Draw the solution in, above the mark on the neck of the pipette. Quickly remove the pipette bulb and put your thumb on the end of the pipette. Release pressure from your thumb very slightly so that the liquid level slowly drops. Once the bottom of the meniscus has reached the calibration mark on the pipette, press down firmly with your thumb. Transfer the pipette to the conical flask and remove your finger from the pipette. Put the tip of the pipette in the conical flask and drain sodium hydroxide solution into the flask. Allow the solution to drain at its own rate. Once it stops draining, touch pipette tip to the side of the conical flask. Do not blow out the remaining solution. The pipette has been calibrated with the last drop left inside. Take the burette initial reading. Record it to 2 decimal places. For example, 10.90 cubic centimeter. The initial reading does not need to be at 0. Place 25 cubic centimeter of sodium hydroxide solution in a clean conical flask. Add 3 drops of phenolphthalein. Note the color changes. Place the conical flask on a white tile so that any color changes can be observed. Then place it under the burette. Carefully turning the stopcock, add small amounts of hydrochloric acid to the flask. Continuously swirl the flask to completely mix the 2 solutions. Continue additions until 1 drop of titrating solution causes a color change which lasts for at least 30 seconds. This is the endpoint of the titration. Record the final volume reading of the burette in your notebook and calculate the total volume of titrating solution that has been added. Record the initial and the final burette readings. Then, complete the observation table. Click the check button when you are done. Now, calculate the average volume of hydrochloric acid added. There is another method to determine the endpoint of a neutralization reaction. Other than acid-base indicator, we can use a pH meter attached to a computer interface. Click on the button to learn how it is done. Other than the normal apparatus like pipette, burette, a beaker, and a retort stand, you need to have a pH meter, a computer interface, and a magnetic stirrer. Prepare the normal setup for a titration. Then, place the beaker on a magnetic stirrer. Dip the pH meter probe into a sodium hydroxide solution. Use a computer interface to link the pH meter to a computer. Switch on the magnetic stirrer and set the computer to record and display the pH. Adjust the acid flow from the burette into the beaker, drop by drop. Record the pH for every 5 cubic centimeter and stop when the total volume of hydrochloric acid added is 30 cubic centimeters. These are the data gathered from the acid-base titration using a pH meter connected to a computer interface. From the data collected, a graph of pH value versus time can be plotted. Click on the Graph button and transfer the data onto the graph. What information can we get from the graph? The pH value of sodium hydroxide decreases as more and more acid is added. At the point where all hydrogen ions are neutralized, the pH plunges sharply. From the graph, can you determine the endpoint? Click on a point along the line. The endpoint is here, at the midpoint where the pH sharply decreases. Can you guess why there is a sharp pH drop? Yes, at that point, there are no hydrogen ions or hydroxide ions left, since all of them have combined to form water molecules. A drop of titrant after that point increases the number of hydrogen ions. Thus, the pH value keeps on dropping. There's another method to determine the endpoint of a neutralization reaction. We know that acid and alkali are electrolytes. That means they dissociate into ions that conduct electricity. And the electrical conductivity can be measured during titration. Therefore, the endpoint can be determined. Click on the button to know how it is done. This is the setup. Let's label the apparatus and materials needed. Drag and drop the label to the appropriate boxes. Put 50 cubic centimeters of hydrochloric acid in a burette and 25 cubic centimeter of sodium hydroxide in a beaker. Then, prepare a normal setup for a titration, an electrolytic cell. Place a beaker of sodium hydroxide on a magnetic stirrer. Dip the carbon rods into sodium hydroxide solution. Close the circuit. Switch on the magnetic stirrer and adjust the acid flow from the burette into the beaker drop by drop. Record the ammeter readings and volume of acid added. From the data collected, we can plot a graph of electrical conductivity versus volume of acid added. At first, the ammeter readings is at its highest, then gradually drop until there is no reading. As more acid added, the ammeter readings increase. What information can you get from this graph? Click on the boxes. At the beginning, there are many hydroxide ions present. When acid is added, hydrogen ions from the acid combine with the hydroxide ions to form water molecules. Thus, less and less hydroxide ions discharged at the electrode. Therefore, the ammeter readings decrease with the increasing amount of acid added. At this point, the ammeter reading is 0. All hydroxide ions present are neutralized by the hydrogen ions from the acid to form water molecule. As more and more acid is added, the ammeter reading increases gradually. This is because there are excess of hydrogen ions present in the solution. Now, from the graph, can you determine the endpoint? Click on a point along the line. Acid-base titration is used to find the concentration of an acid or a base. To solve numerical problems involving neutralization, you must be able to write chemical equations for the reactions. Then, work out the moles of the alkali using the concentration formula. Next is to look at the ratios of the acid and base in the original equation, the stoichiometry. Lastly, find out the concentration of the acid. Click on the example button. In this example, you are required to calculate the concentration of hydrochloric acid used in the acid-base titration. We are going to use a table to solve this problem. This will help you to see and understand what is going on. Later, you might not need this table. Let's begin with step number 1. Key in the chemical equation. Now, key in all the information given, the volume and the concentration of the solution. Now, Calculate the number of moles of sodium hydroxide in a given volume. Next, the stoichiometry and the calculated moles. Calculate the number of moles of sodium hydroxide. Then, calculate the number of moles of hydrochloric acid. Lastly, rearrange the concentration formula to solve the problem.
Solve the numerical problem involving neutralization reactions. Key in your answer. In this lesson, you have learned that an acid is neutralized when an alkali is added to it. Titration is a quantitative analysis method to determine the volume or the concentration of an acid needed to be neutralized by a fixed volume of an alkali. Endpoint is a point when all hydroxide ions from an alkali combine with hydrogen ions from an acid to form water molecules. There are three methods to determine the endpoint of titration. Using acid-base indicators such as phenolphthalein and methyl orange. Measurement of pH value of the solution using computer interface. And measurement of electrical conductivity of the solution. Numerical problems involving neutralization can be solved by using formula MAVA over MBVB equals to A over B, where A stands for acid and B stands for base. The juice of red cabbage can act as an acid-base indicator. What is the chemical substance in the red cabbage that changes color when pH value changes? What color is the juice in acidic and alkaline solution? Find other natural indicators. Collect all your data in a folio to report details about them. Include where they can be found.